Hello, everyone, and welcome to Trends from the Trenches. My guest today works at the seam where genomics meets the hardware that now runs it. He's one of the rare people who can speak to both sides with real fluency. Ben Busby is the Global Alliances Manager for Omics at NVIDIA. His work centers on the rapid prototyping of software for bioinformatics and precision medicine, and on finding better ways to teach complex subjects at the postgraduate level, both in person and online. Before NVIDIA, Ben spent years as a data scientist at NCBI. He's an advisor to Johns Hopkins and adjunct faculty at Carnegie Mellon University, where he still keeps his hands in his own research. Ben, welcome to the trenches.
Thanks, Eleanor.
I'm glad to have you here. So let's get started with telling the audience a bit more about your background. Can you tell us what pulled you out of population genetics and evolutionary biology and into hardware, software, and NVIDIA?
Well, I did a postdoc in evolutionary biology, and it was awesome. But one thing I realized very quickly is that a lot of people were writing their own scripts. Many of them were essentially prototypes, sort of pushing the limits. But a lot of people were working as individuals. So I got really interested in how people write software together and sort of think as a scientific team. And so really started running hackathons, thinking about how to push the limits of what we could do with genomics, as well as population genetics, et cetera, sort of starting to get into multi-omics and multimodal data even then. And that really engaged a lot of people. People got really excited about that. So I think over the last decade or so, I've spent a lot of time doing engagement of folks with computational biology.
Yeah, I think of you as a nerd to nerd translator. Does that sound fair?
Thank you for that. That's very nice of you to say.
So you're still a working researcher in that ongoing work with Carnegie Mellon. What are you working on right now, and what's at stake there?
So there's two things I'm particularly excited about in the research world. One is thinking about really sort of data federation between biobanks, and that's been really exciting, and that's something I've worked on with Carnegie Mellon quite a bit per se. And then the other thing is I'm really interested in being able to process millions of genomes at the scale of biology, but not just in and of themselves, being able to mix with phenotype, as well as transient data types like transcriptomics and proteomics, really, and really sort of build models that are really specific and answer real world questions in this sense.
So, not a big deal at all. Not a big problem.
It just depends on what your motivations are, right? If you want sort of genomics to inform healthcare, then you need to do work. If you don't care about that, then of course you can work on other things.
So then when did you realize that the infrastructure and the hardware was just as important, or at least similarly important to the biology here?
Well, I think one thing I realized is that, again, there were a lot of individuals doing brilliant work, but there were very few frameworks, and this led to somewhat of a lack of reproducibility, as well as, there was a lack of folks being able to pick up on other people's code bases. And so now, of course, we have agents and models to help us with that. But fundamentally, I think it's important to have frameworks for standardization, particularly when we're talking about fundamental types of data analysis.
Yeah. And now that you're in NVIDIA, you get to see up close the free software and the tools that they release to, enable people to use their chips better. So, how well adopted are these by the community? And maybe you could tell us a little about them.
Well, yeah. So, I think a lot of people are quite familiar with NVIDIA in terms of modeling, for example, protein structure, as well as sort of NVIDIA as a whole with its footprints in AI. But there are still a lot of researchers that aren't aware that we accelerate genomics tools, make those open source. That's called Parabricks if you're interested. And then we have lots of tools for single cell, as well as really just accelerating fundamental data science. If you use scikit-learn, for example, check out Rapids.
Okay. Yeah, and the single cell stuff, like my team has seen how much you can accelerate single cell analysis using some of the NVIDIA tools. That is a real thing there.
Yeah, no, it's a couple hundred times faster. So really you can be talking about up to 100-fold cost reduction if you're doing a lot of analysis. So
Yeah, that
that's
cost reduction and also just being able to do the work in the first place at all. Sometimes you can't wait that long to get your analysis. You have to get it tomorrow.
Yeah, and that's something I'm hoping we'll talk about a little bit later is really sort of being able to push into new fields of analysis, really at this sort of new scale of biology that we're living in now.
So then what about do you want to talk a little bit about like what happens when people don't aren't aware of the tools that are published by other researchers, by companies like NVIDIA? Like what happens to them?
Well, I mean, I think it's always challenging to keep up, right? I mean, this is a battle that scientists have fought for the last 100 years at least, if not more than that, of course. So that said, I think things are obviously accelerating faster and faster. And, I was in a interesting meeting on Friday and, with a large group of people, and they were asking how to keep up on things. I think it's challenging, but I think, really thinking about how we take really sort of mixed data in relatively flexible ways and put it into models. I think that's really important. And I think NVIDIA has pivoted a little bit in the last few years in this sense. We're really starting to distribute sort of libraries and fundamental tools for this kind of thing. I mean, I think in many senses, NVIDIA is really a sort of a picks and shovels company. And particularly now in the biological sciences, we're pivoting towards that, making things more flexible. For example, a lot of people are aware of a platform called BioNemo, where we host a whole bunch of open models. And now we're starting to distribute recipes for those open models, which I'm particularly excited about. And then sort of the vision is to enable the community to pass recipes around for tuning some of these models. And I think to me, that's really cool. The idea that we can enable scientists to sort of really be part of an ecosystem. And I think that's the kind of thing that will really enable awareness of new tools.
Right. I love that. Yeah. And free tooling is what bioinformatics was built on from the very beginning, was people building these systems and sharing them. And then they become de facto standards because they were good. They worked. They did the job. So do you want to talk about like a real workflow change that maybe you've seen where, new tooling just like made a huge difference? Maybe some of your CMU work?
Sure. So I've been working on sort of a side project, and you can find it at the haploblocks.org website. It's something that I think is particularly cool, and it's really just a local ancestry tool. So what it does is it puts SNPs and other variants in a local genomic context. And so I think this is particularly important because local genomic context could be quite nuanced. For one, say 50 to 100,000 base pair chunk of the genome between recombination sites, you might be Portuguese type 4 on one copy and German type 8 on the other copy. We've simply built a software to elucidate that. So then what you can do is take SNPs in their particular genomic background and mix them with other SNPs in their genomic backgrounds and then put those into models. So, and I think this is particularly important when we look at complex diseases, and particularly complex diseases in admixed individuals. And we're all admixed to some extent.
Right. And so this is something, is this something that any like individual could use? Like, well, okay, let's say an individual who codes like me. Could I put my ancestry information in there and just get a readout, or put my SNP data from a SNP chip or something in there and get a readout on like where all my blocks are?
So it depends on what you mean by a readout. Like, yes, in theory, you could, I mean, especially if you can code, you could certainly put your own genome in there and then compare to 1,000 genomes.
Yeah.
And you
Yeah.
could see, what your background looks like in the context of 1,000 genomes. And then you could take all the SNPs that you have in ClinVar and see, how those SNPs are contextualized, et cetera, et cetera.
Oh,
So
I
that
like that.
would be, that's something that you could do that might be interesting. But I think, again, the more interesting thing you can do is then you could take those and we have a way to translate these SNPs in context or just the context themselves into binary strings. And those are really easy to put into models. So that's something that we're particularly excited about. And one thing that I don't think we have a whole lot of time to talk about, but we're really interested not only in using this for humans and understanding human biomedical things, but also agriculture. So thinking about taking these types of approaches to plants as well.
That would be really incredible, actually. So it can deal with the multiploidy and the complexity of plant genomes. Is that where you came up with this system in the first place? Is because plants are really difficult?
Well, sort of both at the same time. So we've had to make a few modifications in collaboration with a company called Veil Genomics to think about sort of the nuances of plant genomes, but also, actually, this is a quote from somebody at Veil. He says, for humans, genome grafts are the future. And I believe that. But I think for plants, he said, grafts are the now. And so that's something that kind of resonated with me. And interestingly enough, I mean, what we're doing with these hashes is sort of making approximate dimensionally reduced graphs. But we're seeing that with graph structures all over the place, not just in genome graphs, but in knowledge graphs. And I think that's sort of something that, we're sort of moving towards in kind of an information theory sense in biology.
And you've been a knowledge graph advocate for a long time. I remember that.
I'm a particular fan of knowledge graphs, and I think a lot of people think that they have a particularly sort of pointed relevance now in the age of models.
Okay. Well, for people who are interested, the Haploblocks is published on haploblocks.org. You can check out the paper and the data there. So then, okay, moving on, let's talk about bottlenecks. Every time a new technology is advanced, the bottleneck just moves. It moves somewhere. It doesn't disappear. So where would you say the bottlenecks are in genomic workflows today? Compute, data movement, something else? What do you think?
I'd say it's data, and particularly longitudinal data right now. So, for example, I can make a really cool knowledge graph, for example, with the haploblocks and whatnot, or hash things into a model with haploblocks from genomic data. But what I really want to do is be able to layer on, using a model, transcriptomic, proteomic imaging data, and getting sort of reliable longitudinal data for groups of individuals, typically in a disease-specific way, is a very challenging thing to do right now. And that's one of the reasons I'm so interested in data federation from biobank style data, because I think where we really want to be is thinking about being able to move data from one hospital to another, from one cancer center to another, from one biobank to another, but not raw data, being able to move weights and embeddings in models such that we can have more power for this type of specific longitudinal analysis.
Okay. And so those federation decisions are made, by more often by leadership teams than by the scientists who want to access the data. So where do you think these leadership teams are making good decisions about, connecting science to the hardware, about investing in infrastructure, investing in federation? Where do you think people are doing the right things, and where do you think that they're really not very much?
Well, I think there's an onus on the scientific and technical community first. Now, I don't want to sort of, wash everything with rose-colored glasses and say, if we build a scientific infrastructure, then they will come. I don't think that's true. There still needs to be driving forces from a bunch of places. That said, one of the reasons I'm so excited about the NVFlare platform that I work with a lot is because basically we can make the model embedding simple enough such that sort of the policy people, the legal people, that think hard about the social and ethical aspects of distributing, model weights, model embeddings, can understand exactly what's going in there and understand exactly what facets of the data are being shared. And that, to me, is really exciting. And there's other people that I work with, that other people work with, doing some really cool stuff with latent space representation, same basic idea. So I think what we need to do is present a range of viable options to the folks that do the sort of legal and ethical type of stuff with the biobanks and also hospital data, But I think there's another onus upon us, which is also to show them that there's a there, right? And this is the biggest thing that we are missing in biomedical science is showing that we can build effective models. And we're starting to see really effective models come out of a number of shops. They're being licensed to pharma, they're being licensed to diagnostics, they're being developed by diagnostics companies. But I would say often these models come from counterintuitive places. So, for example, often we see small disease-specific models working very, very effectively. And I think this is something that is somewhat specific to biomedical science and is kind of counterintuitive to some sort of prevailing logic that, sort of longer context, larger and larger models are always better. So, and that to me is really interesting. But when you go back and you think about the genomics of disease, when you think about the genomics of something like schizophrenia versus cystic fibrosis, those are really two very different diseases on a genomic level and may require a fundamentally different model structure.
Right. So you're saying that the one size fits all foundational models that are supposed to model, quote, diseases don't work. It's that we have to be specific about what you're trying to model.
I would say that I am saying that, but not so deterministically. I think probabilistically, there's most of the evidence is pointing to the efficacy of swarms of small models, but I am not saying it's impossible that in 6 months, Anthropic will blow us all out of the water and we'll be working on something completely different because they will have figured all of this out.
Good luck to them. That would be terrifying, but useful. Yes.
There you go. Yeah, terrifying but useful is a really great way to say it.
Amazing. So then where do you see, let's take the opposite thing, some of these tools potentially really powerful. Where do you see it happening that access to better tools is not translating into better outcomes?
Oh, that's a great question. And I think the answer is, and this is almost an old adage, but when people use fancier and fancier tools to ask the same questions, they've been asking for a long time. And I think really that happens when people fail to do the work to contextualize biology, right? If you have a particular SNP in ClinVar and that there's a penetrance measured in, a particular population, in Germany or the United States or something like that, one thing that has been very clear, particularly with papers just in the last few months, is that may not be the same level of penetrance. You may not get the same presentation with folks that are in Pakistan, for example, at least on a sort of societal level. And then I would say that, we often completely ignore, things that have, 2, 3, 4, 5 genomic loci that are really, important in, manifestation and presentation of disease. So I think those are things that we really need to spend time thinking about and do the work to address.
Okay, great.
Are you enjoying the conversation? We'd love to hear from you. Please subscribe to the podcast and give us a rating. It helps other people find and join the conversation. If you've got speaker or topic ideas, we'd love to hear those too. You can send them in a podcast review.
So then sometimes what we see is that companies will buy huge server farm. They'll buy a whole bunch of GPUs. They'll license a whole bunch of time on clouds and provide their teams suddenly a bunch more compute than they had before. So when that happens, what actually changes for them? Like, do they actually make good decisions about how to use that? What are you seeing?
Well, so as the scale of biology changes, right, people often want more and more compute. And typically we don't see researchers getting flooded with compute. I mean, if you're in an academic institution and they buy a bunch of GPUs, that's great. But if you as a biologist don't sort of lay your claim to some of them, your physics colleagues are probably going to gobble all of them up. So that said, I think it's important to do creative things, right? With this amount of compute, we're able to do things like look at 5 genomic sites simultaneously and integrate phenotype and integrate proteomics. And these are the kinds of creative things we should be doing with the new compute we have, right? I mean, data preparation is one thing, and that is super important. And data generation is going to be a huge bottleneck. So we're going to need automated labs for that kind of thing, et cetera, et cetera. But beyond sort of data generation and preparation, which should be really reproducible, we need to really be creative about the kinds of questions we're asking. And we really need flexible frameworks to be able to ask those questions. And I'm just going to be blunt about it. I mean, when I see either academics, startup companies being really successful, they usually have a small amount of on-premise compute and then an ability, or in the case of a large academic institution, a fairly large amount of on-premise compute, and then being able to burst up into cloud using small cloud providers as well as large cloud providers. And so I think really being able to diversify compute portfolios, thinking about what you need, having a strategy there, talking to folks about your IT strategy, really doing the math, doing the homework. That's what is going to make sense. And that's what's going to make a competitive advantage for, say, diagnostics companies moving forward.
So what I'm hearing from you is that science is not dead and we still need scientists. Would you say that that's true?
Yeah, no, I mean, we really need people to ask creative questions. Again, if you're just kind of phoning it in at the end of the day and you're doing technical things with GPUs, but you're not asking the questions that are pushing the envelope in science and biomedicine, you're not changing the world, right? And so I think that's the thing. We need scientists to ask creative questions. And then we also need to figure out how to help models communicate with professionals in the field. And nowhere, I can't think of any other field where that's more important than biology, both medicine as well as agriculture, because you have experts in the field, in the case of agriculture, literally. But in medicine, you have people that have been oncologists for 40 years. They have a lot of model weights in their head, right? And so figuring out how to translate the brain of somebody who's been an oncologist for 40 years into a model, I think, is one of our next frontiers that we want to be working on.
Wow. Okay. That is a big project. How would you even start with that?
So, well, for one thing, I think we need really better software interfaces for professionals who are non-technical to work with models and ways to collect those sorts of weights. And those are things, some people in the community are working on, we're working on, but I'm really excited to see where that field evolves.
And when you say different interfaces for folks, are you talking about the chatbot LLM interfaces, or do you mean something else?
Well, I mean, I think chatbot LLM interfaces were a watershed moment, right? I mean, it got sort of the world and the internet, a lot of sort of bulk human knowledge, sort of into the world of AI. We're going to need really sort of more nuanced interfaces for thinking about how to get medical professionals, agricultural professionals, other scientific professionals to get sort of their knowledge into these models. And they're probably going to be sort of small, specific models. So we won't be able to, they're more likely to be small, specific models. So we won't be able to rake information. We'll have to be very selective about information, and then also do pruning on information that's not relevant. And that's another thing that we really need to be putting some work into, and something that's enabled specifically by knowledge graphs.
Okay. So it sounds like that you think that, the future, the next 3 to 5 years, maybe knowledge graphs are going to be a big player.
Yeah, I think knowledge graphs are likely to be a very big player. I mean, they're really a sensible way to organize relatively static biological information and sort of give checkpoints with models, as well as both validate and prune information. One thing that I'm working on very actively is the idea of democratizing knowledge graphs. I think, it's really important to enable biological researchers everywhere to build knowledge graphs. And we're actually running a competition with the AWS Open Data Program in the fall on building agents to build knowledge graphs. So if that's something you're interested in, please keep an eye out for that. It'll be all over social media, etc.
And what is the name of that again? Can you say that again?
Thanks, Ellen. That's called the BioAgent Knowledge Graph Construction Challenge.
Okay, great. All right. So then let's say you're advising a biotechnology leadership team for a new startup.
Yeah.
You want to tell them, like, what do you tell them about their computing strategy? What is the piece of advice that is different now than it was, say, a year, 2 years ago that you would give them?
Yeah. So for one, you need a computing strategy. And two, I think it's important to do the math. But as I'll say again, I think companies are most successful when they have some on-prem balanced with being able to burst onto cloud. This is where we see people being very successful and being selective about what they buy. For example, in the biological sciences, the RTX 6000 chip is amazing. It does so much of all of this stuff we talked about, from processing of genomics data, being able to run models, et cetera, being able to do some tuning, all of that stuff. That's fantastic. So, for example, you could have sort of a workforce, 4 to 8 RTX 6000s. And then if you need to do some training of a model, you'd want to burst up onto cloud. But I think there's something more fundamental here, which is, so we're starting to see this emerging economy of licensing models, right? If you look at, big business news, et cetera, you see, large companies signing licensing agreements with model building shops. So what I would say to startup companies is, even if you plan to keep the whole thing proprietary and develop a drug yourself or whatever it is, build models that other people would want to license, or build models that you would want to license. Really, you should be asking questions that other people will be so excited about in terms of them moving the needle that they would be willing to pay you for them.
And so that implies that you understand the landscape of the science well enough to know what's missing.
Absolutely. Again, I think maybe this is a little bit, iconoclastic or something, but in fact, I think for computational scientists, right, the next 3 to 5 years is going to be an incredibly busy time because we're going to be working on a lot of these things. We have a lot to work out in terms of doing modeling, but more importantly, validating models, right? And that comes with characterizing benchmarks, and that takes a lot of scientific work to come up with benchmarks that make sense. And really, at this point, to benchmark models specifically and effectively, we need clever biologists who can come up with benchmarks that not only make sense, but also push the limit of science. And that's a really tricky thing to do, right? Not just come up with benchmarks for the science we did 10 years ago, but come up with benchmarks for the science of the future.
And I think one of the things that is really important is understanding that there's also other, we talk about models, there's old technology that still works. And one of the baseline things we need to do is to make sure that any new model that is computationally expensive and hard to make is actually better than the existing models that we have. This is a beef of mine, is that people jump straight to a complex neural network when an actual linear model would work just fine. And a lot of the time, my team has done these benchmarks showing that that's the case. And so I'm super into benchmarks as well. I agree completely.
So I would say, XGBoost is still alive and well. And at the beginning, I mentioned this Rapids thing. Check out XGBoost on Rapids. It's about 45 times faster on GPU than CPU. That said, just to echo your point, we need benchmarks that will compare a linear model versus XGBoost plus a small gen model, or versus a small gen model versus a large gen model, right? You should absolutely be able to compare apples to apples there and say, I'm going to use the most efficient model that allows me to push science forward.
Yeah, absolutely. Okay, we're on the same page here. I love it.
Yeah.
So then what do you wish every bioinformatician now understood about the latest hardware and infrastructure that could change how they work. I think you touched on it a bit already. Maybe this is a little redundant.
What do I wish every bioinformatician understood? I mean,
Yeah.
I would hope that every bioinformatician would think about how to take, and I think that many are, how to take multiple complex data types, and use them in concert to be able to peel the onion of a scientific problem, right? We know that all ICD-10 codes typically are umbrellas for a whole variety of etiologies that lead to fairly consistent symptomology, right? And so being able to come up with a model that starts to peel those layers of the onion back and point towards pipelines where things are treatable, I think that's where I wish every bioinformatician's head was at.
Yeah, makes a lot of sense. I don't. What do you think of my hypothesis that the rise of coding agents to assist in coding just means that bioinformaticians get to be biologists again?
Wow, I love the question. You know, I saw this quote somewhere on social media that I thought was great, and I'm sad I don't have attribution for it. How useful you think language models are really often depends on how much you code. And I think that's something that I've seen be consistent. And so many of my friends who are professional, like, I mean, PhD computer scientist type people have just dove in, right? They love this stuff. I love this stuff. Oh my gosh, it's so great. Being able to prototype so fast. And honestly, I think you're absolutely right. And I would go even a step further and say it doesn't even allow us to be biologists. It forces us to be biologists, because if we can't use all this cool stuff to ask better questions, then the world is going to go work on something else. And, I mean, don't get me wrong. I like animals, but I hope that's not cat videos.
Yeah, that's what I've observed from my team, is that they just get to do more science in the same amount of time. With the coding agents helping them write the code they need to write, they can focus on learning the biology rather than fussing with the details of some irritating syntax of a poorly written language.
Yeah, and I think agents are just going to amplify that trend, right? And so, like, then for basic processes, not the ones that require the creativity I was talking about, not the big combinatorial things, but basic things, right? Like BWA to deep variant or GATK. I mean, we already have agents that wrap Parabricks tools to do that. And so anyone should be able to do the sort of basic foundational bioinformatics processes on their own at this point.
Yeah, well. So then what do you say? One last question, which is, is there something that NVIDIA is building that people don't know about, that everyone is sleeping on, that you think they should know?
Well, there's a number of things, really. One thing I'm particularly excited about from a hardware point of view is the rise of CPUs that go with the GPU, and really there's zero latency in terms of transfer between those CPUs and GPUs. So you really can have your cake and eat it too. And then also those GPUs tend to have a lot of RAM. So you really get deep into biological questions. I think what I'm most excited about in the software sense, although I am hugely biased because I work on it, is the idea of model validation and thinking about, all right, how do we build frameworks that allow people to compete models against each other in a fairly flexible way, such that they know how to compare A to B, whether it's the same model tuned to some extent, whether they're totally different models, et cetera, or different models that specialize in different etiologies of disease.
So if people can run their own pipelines without, necessarily knowing how to code in the first place, like I agree, that is absolutely a thing, but biologists are now much more empowered to do things on their own. So what happens to all of us bioinformaticians who spent so much time learning to code? What are we good for?
Actually, I think counterintuitively, over the last few months, I'm spending more and more time referring companies to small bioinformatics engineering shops because they might have one or two bioinformaticians who can use agents, spin up prototypes, do a proof of concept. That's amazing, right? But for hardcore engineering and efficiency, particularly when somebody like NVIDIA gets involved, and we can give great advice, but our advice is best given to professional engineers. There need to be a cadre of working people that are working engineers, working bioinformaticians who can develop a stable, reproducible code base, particularly in biomedicine, because we are talking at the end of the day about giving reproducible clinical decision support that is used to treat patients.
Amazing. So we have jobs still. Is that what you're saying? There's a use for us?
I'm not saying you have jobs still. I'm saying those jobs should expand.
Ah,
My kids aren't quite old enough yet, but if my kids were, sort of finishing college and thinking about computational biology, I see this as a growth industry.
amazing. Thanks so much for joining me, Ben. I really appreciate you taking the time, and it's been a really interesting conversation. It's so good to hear your perspective.
Absolutely. Yeah. Thanks for inviting me. And I look forward to talking soon and seeing everybody at various meetings over the next year.
It's been a pleasure having you on the trenches.
Thanks a lot.